Welcome to the BioInsights, where science comes alive.
All right, let's dive right into this explainer and demystify the incredible, literally microscopic battles happening inside our bodies right this very second. We hear about enzymes all the time, right? Those tiny biological machines that basically make life possible. But honestly, the really fascinating story is how they're controlled, stopped, or even permanently destroyed by outside forces and internal regulators. Now, you might be wondering, what in the world does a deadly weapon like sarin nerve gas have in common with that everyday aspirin sitting in your medicine cabinet, or, the penicillin you take for an infection? Well, the answer comes down entirely to something called enzyme inhibitors. These microscopic agents have the sheer power to dictate life, death, health, and disease, and today we're going to see exactly how they pull it off. So here is our game plan for today. We'll start with number 1, the biological machines, then move to 2, the competitive blockers, 3, the allosteric saboteurs, 4, the uncompetitive trap, 5, the suicide inhibitors, and wrap up with 6, medicines, poisons, and feedback. Let's get right into it. Part 1. The biological machines. Setting the baseline. Okay, so think of every enzyme as having a primary VIP parking space. We call this the active site, and it's meant for normal substrates to bind. This is where the actual heavy lifting gets done, like breaking down the carbs or proteins from your lunch. But, and this is crucial, a lot of enzymes also have a sneaky secondary backdoor called the allosteric site. This spot isn't for the substrate at all. It actually acts as a structural control switch that can completely warp or change the enzyme's overall shape. To really grasp how inhibitors work, we need to look at 2 main metrics that measure an enzyme's performance. We've got Vmax, which is its absolute top speed and capacity, and Km, which measures its biological need or affinity for a substrate. Think of Vmax as the absolute top speed of a sports car, and Km as how eager the driver actually is to get in that car. A higher Km means the enzyme actually has a lower affinity. It basically needs a much higher concentration of substrate floating around just to get the reaction going. Keep these 2 vital metrics in mind because they're going to be absolutely key as we introduce the inhibitors. Moving on to section 2. The competitive blockers, reversible rivals. Imagine a microscopic game of musical chairs for that active site. A competitive inhibitor is shaped incredibly similar to the normal natural substrate. Because they look so much alike, the inhibitor just sneaks right into the active site and blocks it. But they don't lock the door behind them. It's like a seesaw. If we just flood the system with enough natural substrate, it shifts the balance and literally forces the competitive inhibitor right out. It is purely a numbers game. And hey, this isn't just some textbook theory. This is exactly how some of the most common everyday medicines work. Statins, like Lipitor, lower your cholesterol by physically competing for the active site of a specific enzyme. ACE inhibitors do the exact same thing to reduce high blood pressure. They just sit right there in the active site, so those harmful biological processes are slowed way down. Because the natural substrate can eventually outcompete the inhibitor, if we just dump enough of it in there, the maximum speed, our Vmax, remains beautifully unchanged. The enzyme can absolutely still hit top speed. It just takes way more substrate to get there, which means our Km, the required concentration, goes up. All right, section 3, the allosteric saboteurs, non-competitive inhibition. Now these guys play by completely different rules. Unlike our last group, non-competitive inhibitors don't even bother fighting for the active site. They use that back door we mentioned earlier. They bind to the allosteric site and literally warp the whole machine from the outside. Because the active site itself gets fundamentally deformed, adding more natural substrate won't help you at all. It just won't fit anymore. So that whole seesaw trick, yeah, totally useless here. For a real-world look at this allosteric sabotage, think of certain antidepressants called MAOIs, which alter the enzymes responsible for breaking down neurotransmitters. Or oxamic acid, which actually stalls a key metabolic pathway related to glucose by, you guessed it, warping the enzyme's structure. So here's the kicker. Because this saboteur isn't fighting for that main parking space, the enzyme's innate affinity for the substrate, the Km, stays completely unchanged. The substrate can still try to bind to the unwarped enzymes just fine. But since a whole chunk of your enzymes are permanently warped and basically taken out of commission, the overall maximum speed of the system, the Vmax, drops dramatically. Next up, section 4, the uncompetitive trap. Lock it down. This scenario is just wild. Uncompetitive inhibitors are uniquely patient. They don't just randomly bind to the free enzyme, no. They wait until the natural substrate is already perfectly seated inside the active site. Only then does a specific little allosteric pocket open up. The inhibitor slips in and boom, locks the door from the outside. The substrate is literally trapped inside and absolutely zero product can be formed. We see this unique trapping mechanism do some severe damage with things like arsenic poisoning, which binds and just halts critical cellular energy production. But incredibly, we actually use this same trapping mechanism therapeutically too. Lithium, which is used to treat bipolar disorder, acts as an uncompetitive inhibitor within very specific cellular cycles in the brain. Before we get into the really irreversible damage, let's quickly summarize exactly how competitive, non-competitive, and uncompetitive inhibitors alter our key metrics. Notice how uncompetitive drops both Vmax and Km. Basically, if you want to understand exactly how a drug or a poison is acting in the body, checking these two metrics tells you exactly what kind of microscopic battle is taking place.
Section 5. The suicide inhibitors. The assassins.
Okay, we are entering the permanent zone now. The secret to this level of destruction is the formation of a covalent bond. While those reversible inhibitors use weak connections that can eventually break apart, suicide inhibitors use covalent bonds, which are basically a permanent molecular superglue. The inhibitor binds, acts like a normal substrate, and then irreversibly fuses to the enzyme. That enzyme is permanently dead. It will never function again.
This total, permanent shutdown is exactly what makes SARM nerve gas so instantly deadly. It irreversibly shuts down the enzymes controlling your nervous system. But get this, we actually harness that exact same assassination tactic for our own health. It is literally how aspirin stops inflammation, by permanently binding to COX enzymes. And it's how penicillin safely destroys bacterial cell walls to cure infections. One single mechanism acting as both a devastating weapon and a complete miracle cure.
And finally, Section 6, medicines, poisons, and feedback. Let's bring this all together and see how our own bodies act as the biological cops. It's called feedback inhibition. Imagine a natural assembly line passing a product down the chain. When you finally have enough of the end product, that final product loops all the way back around and acts as a non-competitive inhibitor on the very first enzyme in the chain. It just warps the starting enzyme's allosteric site to shut off the whole assembly line. It perfectly prevents wasteful and potentially harmful overproduction. Honestly, our biology is just brilliant. So, knowing that we can manipulate these microscopic battles with everyday medicines or even deadly toxins, what future diseases do you think we're going to conquer next by mastering enzyme inhibition? I mean, we've already tamed cholesterol, managed blood pressure, and conquered bacterial infections just by blocking enzymes in creative ways. The possibilities for the future of medicine are absolutely boundless. Thank you so much for joining me on this explainer, and I'll leave you with that exciting thought to ponder.